Okay, well, thank you everyone for showing up. I want to thank the organizers for inviting me. And the last time I was at PSA was probably 20 years ago when we were sharing rooms at college campuses and dormitories. I don't know if anyone remembers that, but we've come a long way. So, let's see. So I'll just briefly go over things that most of you probably know anyway, but coccidiosis control depends on a large part on understanding the epidemiology of the disease and the causative protozoan Eimeria. So you might ask, how can molecular biology be used to study and control avian coccidiosis? Aside from Eimeria maxima, most Eimeria species cannot be differentiated from one another microscopically. And this is an example of a slide of oocysts isolated from fecal material. And you can see that when we've done molecular analysis of this sample, we find 4 or 5 different species. But I think you'd be hard-pressed to, except for Eimeria maxima, which is the largest, hard to distinguish the other species. So molecular biology based on sequence differences of different species is very useful. And the reason is that if you look at the average size of the 7 major species of Eimeria, they're very similar, and there's an overlap in the length and the width of those. So it would be easy, or I'm sorry, it would be very difficult, except for Eimeria maxima, to distinguish these because of the size overlap. So why is it important to know the concentration and species composition of Eimerian litter? And there's multiple reasons that this could be used. An overabundance of a single Eimeria species may reflect drug resistance in that species. An increase in say, Eimeria maxima, for instance, may reflect immunovariability. So if you're using a vaccine and you have a large increase in Eimeria maxima, it could be that immunovariability is at play. And we'll show some evidence for that. An increase in Eimeria oocysts concentrations may corroborate coccidiosis or necrotic enteritis diagnostics. Molecular biology is useful for typing Eimeria strains, such as what is used for avian influenza, Newcastle disease virus, and other viruses. It's just the same technology looking for sequence identity of these species. Another question that could be answered is, are more virulent strains emerging? So if you have a problem on a farm, you could isolate the oocysts and look for sequence patterns that may reflect different populations that are arising that are more virulent. And using that information, is there a correlation with the presence of these strains and the severity of coccidiosis or necrotic enteritis? But a lot of people, especially in the poultry industry, ask, how is this information helpful to us in growing chickens? And there's a number of ways this could be used. One is you could switch coccidiosis controls to drugs if immunovariable strains are rising. You could also use another anticoccidial drug or switch to vaccination if drug-resistant strains arise. And I'll give an example of how we've used molecular biology recently to understand Eimeria oocysts population dynamics in a poultry house. As many of you may know, companies place auxiliary feed lids in the poultry house, in a broiler house, for about 7 to 10 days after placement of the chicks. And the reason is that they're trying to encourage chicks to start eating soon after placement. And these auxiliary feed lids are interspersed with fecal material because young chicks can easily walk in and out to access the feed. And it occurred to me while we were sampling litter that these lids had fecal material all spread throughout the feed. And there's a contrast between the fecal material and the feed. So it makes sense that the birds would actually be attracted to those fecal material and possibly ingest oocysts. So the question was, does the fecal material in the auxiliary feed lids contain Eimeria oocysts? And if so, are these oocysts viable or infectious? Because this could be a source of Eimeria oocysts to young chicks. So we took fecal samples from the feed lids and from litter in the house, and we processed them for Eimeria oocysts. We did oocyst counts on the fecal material in the feed lids, and we did PCR analysis to try to identify the different species that were present. And we inoculated susceptible chickens with these oocysts. We didn't sporulate those. We just took those to see if they were actually capable of causing infection. And what we indeed found was that Eimeria maxima oocysts were present in the feed lids and the litter from all the broiler houses. And we corroborated this with PCR and oocyst counts. So the dark is the oocyst counts. And these are essentially 2 houses on 3 farms, either feed lids or litter. But the bottom line is that we found Eimeria maxima oocysts virtually present in all the feed lids and in the adjoining litter. We also found Eimeria cervulina, mitis, praecox, and tenella present in the feed and the feed lids. However, what we did find was that only Eimeria cervulina and Eimeria mitis oocysts that were isolated were actually capable of causing infection in susceptible chickens. And those of you who have worked on cocci for a long time will know why it was only A. cervulina and mitis that were causing infection, because they're excreted. Their patent period is much shorter than the other species, Maxima, Tenella. But also they sporulate. Because they have a head start, they sporulate much more rapidly than the other species. So how molecular biology was useful here is that we not only showed that there was a source of infection in these feed lids, but we could identify which species were present. And now we can recommend to poultry producers that if you're going to use feed lids and you're on a vaccine program, you need to get the feed lids out of the house by 6 days, or you may have Maxima and Tenella sporulating and causing infection as well. So let's switch gears a little bit. The current methods of PCR analysis to identify species are based on a number of genes, but basically the ribosomal DNA, the DNA coding for ribosomal RNA. And that's the ITS1, 2, or the 18S. It's a very sensitive assay because ribosomal DNA is in multiple copies in the genome. So it's extremely sensitive assay. It's not a single copy gene. But the problem with that, because it's sensitive, it's not quantitative, because different species of Eimeria may have different copy numbers of these ribosomal DNA. My lab is developing a metagenomics approach to determine species composition and the relative numbers of Eimeria species in any sample. And our goal is to use single copy genes to do this, that we can quantitate, we can relate the number of sequences that are identified metagenomics and mapping to the GenBank deposited sequence. This is used by the poultry industry to study the microbiome of chickens, and it's used in human medicine. And you can simultaneously analyze up to 96 samples in a single test tube. And we're using the cytochrome oxidase gene, the mitochondrial sequence, to test this. The hope is that metagenomics can not only reveal species composition and relative abundance of each Eimeria species, but also provide a means of identifying strains. That is, strains that may be immunovariable or drug resistant or highly virulent. In all likelihood, however, I must admit that we'll have to require sequencing the entire genomes of these organisms to identify single nucleotide polymorphisms associated with phenotypic trait. And then the goal is if we can identify those SNPs, then we can develop a PCR-based sequencing method for marker identification. The goal would be that if you have drug resistance, if you have evidence for drug resistance, then maybe you can take a sample and do a rapid PCR method to identify or characterize the drug resistance in that litter. The progress on sequencing so far has been good but frustrating. Eimeria tenella APU1 genome we've got that to the chromosome level. There's 15 chromosomes. That was based on scaffolding, again, using the Eimeria tenella identified by the Sanger group in the UK. Independently, we've done the Eimeria cerevisiana APU1 genome to the chromosome level, and that will be submitted for publication next week. The most important in our mind is Eimeria maxima, getting that to the chromosome level, because we have drug-resistant strains of Eimeria maxima. We have strains that vary in virulence, and it would be nice to use the genomes to compare these to identify markers for virulence or drug resistance. So how are we going to use this information? And I think Hector talked about this earlier, is that why is it that different Eimeria species invade different regions of the gut? They're very site-specific, and can we use the genomes or the genetic information to identify genes that may be responsible, or transcripts using RNA-seq, that might point to the genetic basis for the intestinal site specificity? So basically, we would align the chromosomes of 3 different species of Eimeria and look for differences between Eimeria maxima, tenella, and A. cerevisiana to hopefully pinpoint sequence differences. Maybe entire sequences are absent, or genes are absent in one or the other. It's kind of exciting that you might be able to answer that question that Hector actually brought up. Why is there site specificity? Can we compare genomes of virulent and less virulent Eimeria maxima? We have a Eimeria maxima we've isolated that can reduce weight gain by 25% with as few as 1,000 oocysts, with another strain that requires 10,000 to 20,000 oocysts to get the same. So what is the basis for that virulence? We've developed drug-resistant lines of Eimeria maxima by passaging Eimeria maxima sensitive in the presence of drugs, and we have a number of drug-resistant lines that we're hoping to develop markers. Well, if you have the genomes of the resistant and the sensitive, what's different about the drug-sensitive and drug resistance that contributes or is the reason for the drug resistance? And hopefully we can develop a marker for that. All right, so now I'm going to switch to the second part of my talk in 11 minutes on vaccination. So there's a lot of controversy in the field about this, but the ultimate goal of vaccination, in my mind, is to ensure that every chick ingests a number of Eimeria oocysts sufficient to elicit protective immunity against coccidiosis by 2 weeks of age. I think it's too random, in my opinion, to assume that boosting effect by chicks that have not gotten vaccine would actually be protected, because chances are they're going to come down, depending on how many oocysts they've ingested, will come down with acute coccidiosis and necrotic enteritis early in life. So my goal and the goal that I have for my research is we got to develop ways that gets every chick, or virtually every chick, immunized before it's placed on feed. Early in a chick life, and that is 2 to 3 weeks, coccidiosis predisposes it to necrotic enteritis because of the damage that cocci does to the mid-intestine. And it leads to increased mortality. Later in a chick's life, around 5 weeks and thereafter, it leads to poor weight gain and lower feed conversion efficiency. So we need to get chicks either protected by drug treatment or we need to protect them by vaccination. Why is it important to vaccinate every chick? Well, there's high numbers of viable Eimeria oocysts are present at time of placement and can cause outbreaks in immunologically naive chicks, those not taking up vaccine. We did a study several years ago where we went into poultry houses after the chicks were moved out, and we looked at oocysts that were present in the litter at 1 and 2 weeks prior to placement of the next chicks. And we found that 30% of broiler houses on the eastern shore of Maryland had viable oocysts at time of chick placement. So to assume that the ammonia levels or the heat is actually going to kill all the oocysts, even windrowing or crusting off, is going to eliminate those oocysts is not correct. I think you can assume that these oocysts are not viable. So there's oocysts in the house, they're viable. You're putting chicks in the house that are immunologically naive, and it's just, in all likelihood, it's inevitable that some of these chicks are going to come down with coccidiosis. Although commercial Eimeria vaccines are effective, I will be the last to argue against that when given per os, orally. In our opinion, in our research, spray vaccination is neither uniform nor efficient. And basically, you've seen this slide where a tray of 100 chicks is passed through a vaccinator spray, and they're sprayed with a red dye that's either aqueous or gel. And the concept is that these chicks will preen themselves or each other and thereby ingest oocysts. But anyone that has seen a tray of 100 chicks, most of these chicks are not moving, and they're not very aggressive. There are some chicks that will take up lots of oocysts, more than the infectious dose or the immunizing dose, and they're the ones that, as Don Ritter would point out, are putting out lots of oocysts in the litter. So we had two approaches that we've taken. We have one more that we're approaching, but I won't talk about that today. And one is gelatin beads impregnated with Eimeria oocysts applied to feed. Now, this was done by the UK group years ago using a different technology. And the other that we've developed, this also has been used in a different way in the poultry industry. I'm not claiming that we were the first to do this, but I think we've refined this to make it very practical. So I'm not going to show a lot of data, but what we found is that if you incorporate Eimeria oocysts into gel beads and you put them on feed, you find that the oocyst production in chicks allowed to feed on gel beads containing oocysts produce 10 to the 6th to 10 to the 7th oocysts. By spray vac, we don't see that. We see a much lower output, significantly lower than the gel bead or the oral gavage control. The variability is higher also with spray vac compared to gel beads. And we get nice weight protection in chicks that are orally challenged with the Eimeria maxima or any of the other species in gel beads. So you can see the typical controls, non-immunized gaining 600 grams, non-immunized challenge with the parasite, significantly less weight gain. But with gel beads or with the control, you're getting nice protection against weight gain loss compared to the spray vaccination. So gel beads, oocyst incorporated in gel beads is an effective way to do this. However, as I'll admit, it's probably impractical. We tried this through Don's help. We tried this with 16 million broilers, and the results were very promising. But after doing this for about 2 weeks, the GROAC guys said that I don't know that we're going to be using this technology because you have to manufacture the beads, you have to transport the beads to the poultry farm, and you have to put the beads out on the feeder. and that requires salary, which a lot of poultry companies don't want to expend. So it works, but we have to figure out a way to deliver this economically for the producer to actually use it. The technique that I'm really high on now is vaccination through drinking water. We actually have a patent pending on this, the adaptation that we've come up with for this. So it's really easy. It only requires 2 people. At 3 days of age, you raise the water lines for about 3 hours in a broiler house. Or you can, we've also shown that you can allow the chicks to drink down the water and thereby empty the water lines. You dilute the commercial vaccine, calculating the volume of the vaccine you need based on the number of chicks in the house and the size of the broiler house. You charge the line with vaccine containing water, right, and it has a blue dye that we mix in. So when the blue dye gets to the end of the water lines, you turn off the valve, you lower the lines, and the chicks, because they haven't drank for 3 hours, all run over. They get satiated very rapidly because they're only 3 days old. So they drink enough vaccine, but they don't overconsume vaccine. So the aggressive ones make it over to the waters. They get satiated, leave. The more passive chicks then go over and get their share. So within a half hour, you can get all the chicks vaccinated with the vaccine. We have shown that you can get greater than 90% uptake of the oocyst delivered this way when you measure oocyst output 5 to 8 days. And we have a publication in Journal of Applied Research if anybody wants to see the data. The advantages of water vaccination is that it's highly effective, requires only 30 minutes per house, and the method has already been used or is being used to administer some viral vaccines, such as LT. The only problem with the LT is delivered a little later than the 3 days. So trying to convince poultry companies to combine an LT vaccine with a Cocci vaccine is now the next issue. Disadvantages, requires 2 people, right? Hatchery spray vaccination requires 1 person, and it's at the hatchery. So you need someone to monitor the water lines. You need somebody to introduce the vaccine into the water system. Part of our patent pending is an automated system for introducing this that hopefully will eliminate that need. Conclusions. So molecular biology allows one to study Eimeria oocyst and avian coccidiosis at a more precise level, possibly identifying drug resistance markers and a genetic basis for virulence and intestinal site specificity. Vaccination using Eimeria oocyst and gelatin beads or delivered through the water system has the potential to achieve 100% coverage. But the big question is, will poultry companies adopt this technology? It's only if we can show that we can protect against necrotic enteritis and we can improve performance either through better feed conversion efficiencies or weight gain in that. Because if they're faced with paying another salary and having a vaccinator go to all these farms, that's an expense that they would rather not incur. And I'll take any questions.